The first stage of smear preparation is to write on the ground glass portion of the slide the name or number of the specimen. Pencil is used. It won't wash off during the staining process, as many inks will. Again, the loop and mouth of the bacteria sample container are sterilized prior to smearing. After obtaining a sample, the loop is used to spread it over the slide surface to create a thin smear, as shown here. In some situations, the bacterial culture may be so thick that a drop of water must be added to the sample to dilute it enough to create a thin smear. In others, such as fluid, dilute broth, or urine samples, the culture may be so thin as to require several layers of thin smear for the bacteria to be visible and prevent the smear from being washed away during the staining process. Usually 4 to 6 loops full of sample are needed. To speed drying and fix the smear to the glass slide, it's touched to a flame 2 or 3 times, with the smear side away from the heat. Now you're ready for staining. The fluids used in the staining process are, from right to left, crystal violet, iodine, ethyl alcohol, and safranin. The first dye used is crystal violet, which adheres to the peptidoglycans of the gram-positive bacteria. It's allowed to remain on the smear for about 1 minute before being lightly washed off with clear water. Next, iodine, which provides a mordant or chemical hook to bond the crystal violet to the peptidoglycans, is poured over the slide. The use of mordants was derived from the textile industry, where they were used to bind dyes to textiles. After being allowed to stand for 1 minute, the iodine too is washed off. It's gently washed off with 4 to 5 rinses over a 20-second time period of 95% ethyl alcohol. This halts the mordanting process and extracts any crystal violet which has not been fixed to the smear. Any remaining alcohol is gently washed off with clear water. Safranin is then applied. Safranin is a counterstain. It stains the organisms which could not hold the initial crystal violet dye because they lacked the peptidoglycans of gram-positive bacteria. Therefore, these organisms are gram-negative. On the slide, they're revealed by the pink color imparted by the safranin. After 30 to 60 seconds, the safranin too will be gently rinsed off with clear water. The slide is then dried in blotting paper, also known as bibless paper, and it's ready for viewing.